The innate immune system triggers a fast-acting response when exposed to infection and is considered one of the first lines of defense for the body. Without it, we are left vulnerable to disease. To better understand the innate immune system and its impact on human health, collaborators of the Morehouse Lab sought to identify proteins with previously undefined innate immune function. In their search, they identified a human protein called SIRanc, as it was structurally similar to a bacterial innate immune protein, the SIRanc. SIRanc was shown by our collaborators to be essential to respond to certain types of bacterial and viral infections. The current proposed mechanism of SIRanc activation is as follows. First, upon recognition of an infection, one of the proteins that a cell may signal is SIRanc. SIRanc then likely forms these large, ordered, multi-protein structures called oligomers. Oligomers are held together by the attractive intermolecular forces between proteins. Data from our lab suggests that SIRanc is activated via oligomerization, in which SIRanc oligomers are able to cleave a molecule called NAD+ to execute an immune response. Current literature also supports a similar oligomerization-dependent mechanism of activation for this A. To further investigate SIRanc and the mechanism of its activation, the focus of my project was to specifically determine which amino acids, the molecular building blocks of proteins, were important for the interactions that hold the SIRanc oligomer together. A recent study identified key amino acids of the bacterial immune protein THC that were necessary for THC oligomerization and activation. Given the similarity between SIRanc and THC, we hypothesized that similar amino acids in SIRanc might also be necessary for SIRanc activation. To explore this, we mapped key amino acids found in the study onto matching amino acids of SIRanc. We then generated mutations converting the matching amino acids, which were lysine and aspartic acid, which are charged and able to hydrogen bond, to the nonpolar amino acid alanine to disrupt potential protein-protein interactions. Here, we use a version of SIRanc, where the two tails are truncated off, as they may impede structural analysis further down the line. We can determine the size of the SIRanc structures formed by performing size exclusion chromatography and analyzing the output. The x-axis represents the molecular weights of the structures, with the largest structures found on the left and the smallest on the right. The y-axis represents absorbance at 280 nanometers, which correlates to the amount of SIRanc found at each peak. With wild-type truncated SIRanc, we observe structures with high molecular weights that correlate to multi-protein oligomers on the left. If the mutated amino acids are important for oligomerization, then we'd observe peaks close to the right for mutated SIRanc. Looking at the trace for mutated SIRanc, we observe that the profile is different and shifts slightly to the right, suggesting that the mutated amino acids do impact oligomerization, though it appears that they are non-essential to form oligomers, as mutated SIRanc is still able to form large protein structures, as indicated by the peaks still appearing on the left side of the graph. While there are more mutations we want to explore, these initial experiments will hopefully help build our understanding of structural features that are important for SIRanc to work, which can provide a basis for drug designs that target SIRanc-related diseases, as preliminary data suggests that mutations in SIRanc may lead to the development of certain cancers. Finally, I'd like to thank the Morehouse Lab for their continued mentorship and support. I'd especially like to thank Irene and Emma, who are major contributors to the SIRanc project.